Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the eukaryotic transcription process. Okay, in my previous video I have already explained about the prokaryotic transcription. So here I will just try to explain the eukaryotic transcription process, mainly the initiation and termination. Okay, you just check out the prokaryotic transcription, then the process, how the transcription process occurring, you can understand from that how elongation, initiation and the termination. Here just I try to describe just only the initiation and the transcription, how this is occurring in the eukaryotes. Okay, this is the complex process in eukaryotes. There are many factors and many proteins these are involved in this initiation process, and obviously its termination process is another thing. Okay, and this is totally different from the prokaryotic transcription. Okay, and this is the, here I will try to explain about the process only, and the factors and the proteins and mediators, promoters, these are involved in the transcription process. So this I will try to explain in another video. Okay, because these are too elaborate discussion, so I will try to explain in another video. So here just transcription process is saying that the genes transcribed by the RNA pol 2. Okay, here the eukaryotic transcription process, this is done by the RNA pol 2, and this RNA pol 2 first, this is the first process, first bind with the general transcription factor. General transcription factor, the short form of this is the GTFS. And this GTFS that involved in the transcription from all polymerase 2 promoters and constitutes part of the basic transcription machinery. So here in the general transcription factor, the RNA pol or the promoters, these are involved. And another important thing is that the TF2D, this is the transcription factor 2D. This is also another factor that has the important role in the transcription process. And this is the complex made up of the TATA, Tata binding protein. This is called the TBP protein, and at least 12 TBP associated factors. These are called the TFS. Okay. So here this TF2D is a complex that constituting TBP. TBP means Tata binding proteins, and the 12 TBP associated with associated factors, or the TAFS. And TBP is a sequence that is specific protein and binds to the DNA via its unusual TBP domain. So TBP that bind with the specific protein by the TBP domain. Okay, TBP domain, and which makes contact with the minor groove in the region of the data box. That means before making a complex, there is important role of the TBP, because if the TBP bind with the help of the domain, then there is a role played by the data box. Okay. And here in the bracket, that is only that what is the data box I will try to explain in the another video, but here just it is what that explained very shortly. TBP is the universal factor, and that is required also for the initiation by the RNA pol 1 and 2. So just what is saying that the TBP not only responsible for the activation, or it is involving the activity of the RNA pol 2. It is also involved in the transcription process, where this is activated by the RNA pol 1 and RNA pol 3. And now another thing is that one thing. Okay, so just I was checking the full thing is already is seen from the camera or not. Okay, so what is saying that after the TF2D that has attached to the core promoter. Promoter is one kind of complex, you can say, or you can say this is the factor or another subunit. So first of all, TF2D that bind with the promoter, and here the pre-initiation complex is formed. The short form is the PIC. This is formed. This is called the pre-initiation complex. And attachment of remaining GTPase. So not only transcriptional factor 2D and promoter, they can bind with each other. Another, all the GTPase, that means general transcription factors, these are also bound with the complex and form a subunit, or not subunit, form another complex. And these GTPase bind to the complex in the order. Okay, there are V GTPase that bind with the PIC or the pre-initiation complex, TF2A, TF2B, TF2F, and RNA polymerase 2, TF2E and TF2M. So all these are the GTPase or general transcription factor that bind with the TF2D and the promoter and make a complex. Final step is what? That assembly of the initiation complex is the addition of the phosphate group to the C-terminal domain. This is called the CTD. So complex is totally completed, or complex formation is totally completed when CTD also bind with that. Okay, CTD also bind with that. That means CTD is the C-terminal domain. When bind with this total complex, then the actual structure of the complex is completed. Next is that, okay, another thing is that large subunit of the RNA pol 2 also bind here. Now the CTD contains, now here just only one thing is saying that CTD is the constituents of what? The CTD contains a stretch of severe amino acids. Sorry, severe not, 7 amino acids. Okay, the CTD contains 7 amino acids. So there are the names. These are also the subunit or the amino acid that are present within the CTD. That is okay. Once phosphorylated, the polymerase is able to leave the pre-initiation complex and begin synthesizing RNA. What is saying that? Once the phosphorylated, the polymerase, before phosphorylation, the polymerase never be activated. Okay, when the polymerase is totally phosphorylated, then it becomes activated and then it tries to release from the pre-initiation complex, and now it is activated. In this stage, it is responsible for the synthesis of RNA. Okay, and the phosphorylation might be carried out by the TF2s. So which factor is responsible for the phosphorylation of the RNA pol 2? This is the TF2H. Transcription is a complex here, and it requires the presence of transcriptional activator. Okay, activator is another protein. Without activator protein, the transcription process is not occurring here, which bind to the specific sequences in the DNA, and a typical eukaryotic gene has many activator proteins, and which together determine its role and pattern of transcription. So what is saying that not only the activated activated DNA RNA pol 2, not only the RNA pol 2 is responsible for the transcription process. Here another important role is played by the activator, and there are different activators present, and these are present in different types of transcriptional process, or you can say that the different activators that are responsible for the different transcriptional process in different species. And sometimes it acts from a distance of the several thousand nucleotide pairs, and these gene regulatory proteins help RNA pol, the general transcription factors, chromatin, remodeling complex, and mediator, all to assemble and the promoter. Okay. Another important thing is the promoter. Here is the activator is important thing. Another important thing is the promoter, because without binding of the promoter, the transcription process is not completed. So and mediator also. Mediator and the promoter. This mediator and the promoter. These also play important role in the transcription process. And the mediator allows the activator proteins to communicate properly with the pol 2 and with GT phase. So what is the role of this mediator? Mediator actually it acts as medium. That means between what? Between the activator and the GT phase. GTFS. Sorry, GTPS not GTFS. So what is the role of the mediator? Mediator actually acts as medium between the activator and the GTFS. Okay. So assemble mediator is a general co-activator and of the RNA pol 2 mediator transcription. So this is saying that without mediator, activator and the promoter, these are all the proteins. Without the helping of these, the transcription process in the eukaryotes not possible. That means transcription process in eukaryote is more complex than the prokaryotes. So here the different proteins are engaged in to continue this process. And another thing is I will try to explain about the termination. Okay, so this is the initiation process of the transcription. And another important thing, what type of transcription termination or how the termination process is done in the eukaryotic transcription. So I will try to explain. Okay, so termination process in case of pol 1. In case of pol 1, the gene transcription stopped using a termination factor. Okay, so this is the termination factor, and here a mechanism is just similar like that row dependent termination. And the transcription of the pol 3, here the genes ends after transcribing a termination sequence that includes polyuracil stretch. Okay, polyuracil stretch, and by a mechanism that is resembling row independent prokaryotic termination. Okay, row independent. So first of all, that is pol 1, here the row dependent, and in case of pol 3 there is row independent factor is very important. And in case of pol 2, where the pol 2 is responsible for the transcription, this is the complex process, because here the protein coding genes functionally coupled with RNA processing event. And RNA processing I have already explained, and this is already present in my channel, so please you just check out. Processing event in which the 3' end of the nascent transcript that undergoes cleavage, and after that polyadenylation done. So this is the eukaryotic transcription process and termination process I have explained here, and in the next video I will try to explain the different factors related to the transcription, eukaryotic transcription. So continue with my channel, and thank you.